Thank you for giving me the opportunity to review the process of drug development with small molecules. For this talk, I will be using losmapimod as a case study. These are my disclosures. I believe that many of you have seen this slide before in the other talks that came before mine, and where we see small molecules fitting in manipulation of gene expression is at the level of interacting with proteins that can possibly activate expression. In this case, we are looking specifically at DUX4 gene expression. Before I start, I would just like to give you a brief overview about Fulcrum. Fulcrum is a clinical stage biopharmaceutical company that is using a systematic approach to identify small molecules able to rebalance gene expression. Our vision is to treat genetically defined diseases by addressing their root cause. And we do this by using a high throughput product engine designed to rapidly identify and validate drug targets that can modulate gene expression and treat disease at its root cause. This is our pipeline, and as you can see, losmapimod is our lead and most progressed program, followed by our sickle cell programs, and we also have other programs that are currently in discovery. So some of you may be asking, what is a small molecule? Small molecules essentially are molecules that have low molecular weights. And because they have a low molecular weights, they can enter cells easily. And when they enter cells, they can bind to cellular targets to affect disease processes or affect other molecules such as proteins. Interestingly, nearly 90% of therapies on the market are small molecules. And in 2019, small molecules accounted for more than 70% of novel drug approvals by the US FDA. Some of the characteristics that make small molecules desirable are their route of administration, which is usually oral, and their manufacturing is usually less complex than other types of molecules or therapies. And because we have to think about an oral route of administration, we also have to think about how the small molecule is absorbed or how the drug is absorbed and how it is eliminated and how that will affect our effect on the disease process, as well as we need to consider what is called off-target effects. And this is the result of the small molecule engaging targets that are not intended to be engaged. And usually when we're talking about off-target effects, they usually manifest themselves as side effects. We also have to think about how the small molecule will interact with other drugs and affect the job that other drugs are doing in the body, whether it be blood pressure medications or oral contraceptives. So we need to really think about how these small molecules may affect other drugs, as well as their effect in special populations such as pregnant women and in the elderly. So when we think about drug development with small molecules, we first have to identify a target. And in our case, targets are identified using our proprietary probe library. And our proprietary probe library is called Fulcrum Seek. It's a proprietary database that we use in identifying previously unknown pathway relationships to steer our target identification. But additionally, what also goes into this target identification is knowing about patient-derived relevant models, knowing what it is in our annotated compound library, as well as what is in our CRISPR guide libraries, and also having a familiarity with genetic regulatory pathways as well as genomic databases. So there are many people that are involved in identifying these targets, usually computational biologists, translational biologists, and others to try to uncover these unknown pathways to steer target identification. And once we are able to identify a target, we can then think about candidate development. So in our case, our product engine identified P38 alpha MAP kinase as a key regulator of DUX4 expression. And because it appeared that P38 played a key role in the regulation of DUX4, we then have to validate the effect that P38 has in this regulation of DUX4 expression. And we do that by using genomic and chemogenomic tools across multiple cells. And some of you may be asking yourselves, what is a P38 mitogen activated protein kinase. And p38 alpha beta kinases are members of a family of proteins that modify other proteins and modulate their function in response to extracellular signals. So in this case, the p38 phosphorylates proteins that then regulate that DUX4 expression in the genome and turn it on, essentially. But in order to validate that, we have to take a look at the p38 pathway and its role in the regulation of DUX4 expression. And how we do that is we can potentially look at molecules or compounds that are inhibitors of the p38 pathway. And on the right here in the upper row, you see two graphs, one with p38 inhibitor A and one with p38 inhibitor C. So these are two compounds that inhibit p38. On the x-axis, you can see the concentration of these compounds going from low to high. And on the y-axis, you can see the percent of control. And so we know that the cells that we're using are expressing DUX4, but you can see that in the blue line, we are measuring MBD3L2, which is a marker of DUX4 expression. And you can see that it is going down with increasing inhibition of p38, whether it's p38 inhibitor A or p38 inhibitor C. The gray line that you see at the top that is not changing is a measure of muscle cell health. And because we know that it's important to preserve muscle cell health while we're turning off DUX4, we wanted to also measure this parameter. And you can see that with these inhibitors that there is no effect on muscle cell health. Another way that we can look at how p38 is involved in the DUX4 pathway is by using silencing RNAs. And here at the bottom row, you can see that on the x-axis, there are two rows of control in each of the graphs and two rows of types of siRNAs. And you can see that the siRNAs do inhibit p38 do result in an inhibition of p38 as well as in DUX4. So we can be sure that p38 does play a role in the DUX4 pathway. So now that we've identified our target, which is p38 alpha, and validated this target, we need to model the disease using patient-derived myotubes or muscle cells, and also evaluate compounds that could potentially work through p38 alpha or inhibit p38 alpha to reduce the DUX4 expression. And so one of the p38 inhibitors that we identified was losmapimod. There are several p38 inhibitors that we evaluated as a potential for development for the treatment of FSHD at its root cause. And we did find that p38 did reduce levels of DUX4 in patient-derived muscle cells. And the thinking is that this reduction may stop or prevent functional impairment and accumulation of disability that can potentially enable improved repair of damaged cells. And one of the reasons losmapimod was so attractive as a development candidate was not only because of its properties to inhibit p38, but also because it had been tested in 10 different other indications, and there was a very large safety database. And that safety database showed us that there are few or relatively benign side effects associated with losmapimod in those patient populations that were tested. So we were able to show that the compound that we're looking at does reduce DUX4. In this case, this is losmapimod. We are looking at the reduction of DUX4 in FSHD muscle cells that came from patients such as yourselves. And on the x-axis here, you can see the concentration of losmapimod, and on the y-axis, you can see the percent of untreated. And what we looked at here is we wanted to be sure that losmapimod engaged the target, that target being p38, and how we measured its engagement of the target is by looking at a biomarker called HSP27, and we looked at the ratio of the phosphorylated HSP27 to the total HSP27, and when you have increased target engagement or increased inhibition of p38 alpha, you get a decrease in this ratio. And we saw that as there is increased inhibition of p38 alpha, that there is a concomitant decrease in the expression of DUX4 protein, as well as its downstream transcript or target gene, MBD3L2, and also active caspase 3, which is a measure of cell death. So we were able to see in our cellular models of FSHD that losmapimod inhibits p38, reduces DUX4, the DUX4 target gene, and also prevents FSHD muscle cells from dying. These cellular models can also help predict effective exposures. And in our clinical trial in a dish, we evaluated 11 different FSHD patient-derived cell lines from patients such as yourselves. And these cell lines had different numbers of repeats in them, as well as different mutations, that being the SHMID1 mutation was also included. And what we saw was that there was a 50 to 70% reduction of DUX4 activity in vitro. And when you look at this graph on the right, on the x-axis, again, is the losmapimod concentration, and on the y-axis is the DUX4-driven gene expression. And you can see with increasing concentrations of losmapimod that there is a decrease in DUX4-driven gene expression regardless of the type of mutation that an FSHD cell line has, and that this decrease occurred at 30 and 100 nanomoles, which are clinically relevant concentrations that we can achieve in people. So far, we've identified our target, which is p38 alpha. We've also validated this target using genomic and chemogenomic tools across multiple cell lines, and have also modeled the disease in patient-derived myotubes or muscle cells. And we've identified a compound, losmapimod, that reduces DUX4 expression in our cell models. And at this point, we felt comfortable enough moving into phase 1. In phase 1, we usually evaluate for clinical safety, tolerability, and it can also help with dose selection. So the key questions we asked in our phase 1 program are, does losmapimod show initial evidence of safety and tolerability in FSHD patients? Can losmapimod be detected in muscle at clinically relevant doses? And does losmapimod engage the drug target, p38 MAP kinase, in blood and in muscle? And in this study, in part A, we dosed healthy volunteers with a single dose of losmapimod at 2 different doses, followed by parts B and C in FSHD patients at different doses, and they took losmapimod for 14 days and had muscle biopsies that were done before dosing and after dosing was completed. And what we found is consistent with what has been found in other studies performed with losmapimod, is that the most common adverse effects were headache, dizziness, somnolence, and GI disorders. And as I just said, this is consistent with the large safety database of over 3,500 subjects that have been exposed to losmapimod. Additionally, there were no serious adverse events or discontinuations due to AEs. We were also able to detect drug levels in muscle and similarly in blood, and that these drug levels were dose dependent, meaning that at 7.5 milligrams of losmapimod, here represented in the blue, we see less amount of drug, and we see greater amounts of drug at higher doses. And so here on the x-axis is the dose of the losmapimod, and on the y-axis is the losmapimod concentration in muscle, and this was similar in both muscle and in blood. We were also able to observe target engagement in blood and muscle, showing that losmapimod did in fact inhibit the p38 pathway. And here what we show is in blood is that we show that there is also a dose-dependent inhibition of the p38 pathway. The orange is the 7.5 milligram dose, and the blue is the 15 milligram dose, and it is essentially similar in blood and in muscle. So when you think back to the cell model, you can see that the range of concentrations that we were able to achieve in our phase 1 study are similar to that range of concentrations that did inhibit or decrease DUX4 expression in that cellular model. And so from our phase 1 study did inform safety and dose selection in that we were able to show that losmapimod was generally safe and well tolerated in FSHD patients. We were able to achieve clinically relevant dose-dependent concentration in muscle, as well as show that exposures in plasma and muscle were at concentrations that showed efficacy in preclinical evaluations in multiple labs. And our 15 milligram twice dosing showed sustained and robust target inhibition, thus giving us confidence in bringing this 15 milligram twice daily dosing into our phase 2 studies. So this brings us to our phase 2 in our drug development process. As I said, we've identified the target as P38 alpha. We validated that target. We've looked at this in cellular models. We've also identified a compound that works through the P38 inhibition to reduce DUX4 expression. We were able to look at safety, tolerability, and confirm our dose in our phase 1 study. And now we are at our phase 2 study, which is really clinical proof of concept, showing that how you think your drug may work in the human body is actually what it is doing. So in this case, our phase 2 key questions are, does losmapimod show evidence of an effect on the root cause of the disease, that being DUX4-driven gene expression? And is there evidence of safety and tolerability during chronic dosing? As well as, does losmapimod show an emerging effect on muscle health and selected clinical endpoints? And many of you may already be familiar with our REDUX4 clinical trial that is currently ongoing at 17 sites around the globe, 12 in the United States, 2 in Canada, 3 in the EU. We have completed enrollment with 80 FSHD subjects who are being dosed with either placebo or losmapimod at 15 milligrams twice daily. And our primary endpoint here, again, is our proof of concept endpoint, that being the reduction of aberrant DUX4-driven gene expression in affected skeletal muscles. So there are 2 muscle biopsies in this clinical trial, as well as we are evaluating other endpoints such as MRIs and other clinical outcomes, as well as drug levels and P38 inhibition in blood and in muscle. And so currently, we did have to amend our REDUX4 study due to the COVID-19 pandemic. And we increased the duration of the study from 24 to 48 weeks to allow another opportunity to assess for efficacy. So biopsies can now be collected at week 36 if the muscle biopsy was not able to be collected at week 16 due to the COVID-19 pandemic. Additionally, increasing the duration of the randomized controlled portion of the clinical trial gives us additional opportunities to make more observations about MRI as well as our clinical assessments. And we have retained the ability of subjects that are enrolled in REDUX4 to elect to participate in the open label extension study. So here we are in our journey with our small molecule in our drug development, where we've identified the target P38. We've validated this target. We've modeled the disease in cellular models. We've identified a program that interacts with our target to reduce DUX4 expression. We've shown safety, although safety is a continual journey throughout all of the out of all the phases of development. We've confirmed our dose, and now we are in the process of showing our clinical proof of concept. And if we are able, and when we do show our proof of concept, you then move on to your phase 3 study, in which you want to be sure that when you are, the way that you are affecting the disease process actually garners a clinical benefit for patients. So usually your phase 3 study is larger and has a primary endpoint that is a clinical assessment. So I would like to take a moment and just acknowledge and thank all of the healthy volunteers and FSHD patients for participating in these studies. Our Fulcrum Phase 1, REDUX4, and open label study management team, as well as all of our clinical and scientific advisors and collaborators, the patient groups that have allowed us to interact with patients such as yourselves. We cannot do this without you, as well as our collaborating organizations that certainly have helped in this journey to hopefully a successful treatment to treat FSHD at its root cause. And obviously, throughout this process, takes a lot of people. You saw not only us at Fulcrum, but many others, and especially it takes you, the patients. So thank you very much, and I'm happy to take any questions.